Hi everybody and welcome to our lesson on DNA. We know that DNA stands for deoxyribonucleic acid, and in today's lesson we are going to look at the structure, the location and the function of DNA, and also touch on a few related concepts to DNA as well. So let's jump right into it. We will start off by looking at the location of DNA within a cell. Where is DNA found within a cell? Majority of your DNA is found within the nucleus of your cell. And when your cell is not undergoing a cell division process, in other words, when your cell is not undergoing mitosis or meiosis, all the strands of DNA are in this spaghetti ball-like structure called the chromatin network, which I'm now highlighting in yellow. And if I had to isolate one of those strands, so I'll draw it below this in yellow, we know that that strand of DNA is made up of little building blocks, which I will draw in red. And a certain section of these building blocks is called a gene. So for example, this section might be a gene, and that gene has the recipe or the code which is needed to produce a protein. But let's say, for example, your cell is now going to undergo the process of meiosis or the process of mitosis. When your cell undergoes this process or these processes, DNA replication must take place. So this long strand or yellow strand of DNA has to make a copy of itself. But before it does that, it condenses. So it shortens, it thickens, and it coils, and it forms a structure known as a chromatid, which I've now highlighted in blue. And I will now highlight the structure that is formed when the yellow strand of DNA condenses. It forms one chromatid after it condenses, after it shortens, thickens, and coils. It then makes a copy of itself, a genetically identical copy of itself. And then we have two chromatids, and I'm drawing in the second genetically identical chromatid in red. And these two genetically identical chromatids can then be held together by a structure called a centromere, which I've highlighted in blue. And when two chromatids are held together by a centromere, that then forms a duplicated chromosome. And when we come to the word nucleosomes, that basically refers to the combination of histones and your chromatin network. So your chromatin network is often coiled around protein structures called histones, and that collectively is called nucleosomes. So next we are going to focus on the structure of DNA. So we know from our previous work that DNA is a polymer. In other words, it's a large molecule, and it is made up of two polymers, which are wrapped around each other in a spiral shape. And that's where the word helix comes from. And that spiral structure wraps around itself again. And that's why we have a double helix. If we have to now unwind the structure or uncoil the structure, we will be at this image on this slide. So now let's explore some further aspects related to the structure of DNA. DNA, like we said, is a polymer. Because it is a polymer, it must be made up of smaller building blocks. And these smaller building blocks have a special name, and they are called nucleotides, which I've highlighted in green. So in this little box, we have one nucleotide, which is the building block of DNA. And furthermore, a nucleotide itself consists of three different parts. Firstly, it consists of a phosphate group. It also consists of a sugar called a deoxyribose sugar. And on that sugar, we have an oxygen molecule found in this region. And then our last part of our nucleotide is a nitrogenous base, which in this case is cytosine. However, it can also be thymine or guanine or adenine. We also know that a DNA molecule consists of two DNA polymers. So I'm going to highlight the polymer on the left in a red color, and then I'm going to highlight the polymer on the right in this green color. So let's focus on the polymer in the red block. We can see that your sugar is bonded to a phosphate, and that phosphate is then bonded to the sugar of your next nucleotide. The sugar is bonded to a phosphate, and this phosphate is bonded to the sugar of the next nucleotide. And this bonding is a very strong bond. It's a covalent bond, and it forms the backbone of a DNA molecule. And we see the same thing in your other strand of DNA as well. The strong covalent bonding, which provides your DNA with its support. Furthermore, we can see that these two DNA strands, the red strand and the green strand, as I've color-coded them, bond to each other in certain ways. Adenine always bonds to thymine, or thymine always bonds to adenine, and guanine always bonds to cytosine, or cytosine always bonds to guanine. And these pairs are called base pairs. And because of adenine always bonding to thymine and cytosine always bonding to guanine, we have a law called Shoggoths Law, which states that in any molecule, the amount of adenine molecules will equal the amount of thymine molecules, and the amount of cytosine molecules will always equal the amount of guanine molecules. Lastly, focusing on the structure of DNA, if we look at the red strand of DNA, we can see that the sugar molecules are pointing in an upward direction. And if we look at the green strand, the sugar molecules are pointing in a downwards direction. And so the direction of the polymers is opposite. And the term that we use for that is anti-parallel. And when we speak about the term anti-parallel, it makes more sense for us to use the terms 3' and 5'. But in order to understand this, let's have a look at this nucleotide on the left. So I've already drawn in our oxygen molecule. So remember that your pentose sugar is a 5-carbon sugar, so we need to now find 5 carbons. So we've got our oxygen molecule. Here is carbon number 1. Here is carbon number 2. Here is carbon number 3. Here is carbon number 4. And carbon number 5 is found outside of your ring at this little point here, which then attaches onto your phosphate group. And if we come to the green coded DNA strand, we can apply the same idea. Here we have our oxygen molecule, carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. And so back to the red DNA molecule, we can say that your 5 carbon is going in an upwards direction. And if we come back to our green molecule, we can say that our 3 carbon is going in a certain direction. And that is where the idea of 5' and 3' directions come from. So when we say DNA is anti-parallel, it basically means that the one strand is moving in the 5' or the 5 carbon direction, or the upwards direction of your sugar molecules, and the other strand is moving in the 3' direction, or the 3 carbon direction, or in the downward direction of your sugar molecules. Okay, so as we can see, we understand quite a lot about the structure of DNA, but that was not always the case. Many years ago, scientists were trying to understand what DNA was, and at first scientists thought it was actually a protein. Many scientists underwent experimentation. Scientists such as T.H. Morgan, scientists such as Hershey and Chase, and we also have scientists like Frederick Griffith. And essentially what their experiments concluded was that DNA was not a protein, it was in fact genetic material. A scientist by the name of Franklin, or a full name, Rosalind Franklin, was able to create what is known as an crystallography X-ray diffraction, which is this structure on the right, which basically is an X-ray of a DNA molecule from the top view. So here's your one strand from the top view, and here's your second strand from the top view. And using this image, a scientist by the name of Watson was able to determine some things about the DNA molecule. Firstly, that it was helical. Obviously, because we could see two strands wrapped around each other in the X-ray crystallography, we knew that it had to have been a spiral shape. We were also able to determine the width of the helix, and we were also able to determine the spacing of the nitrogenous bases thanks to the work done by Watson. And again, the fact that there was width confirmed the idea that DNA was in fact made up of 2 strands. So although we know quite a lot about the structure of DNA, it took a scientific journey of experimentation to get there, and we have many scientists to thank for that. So at the beginning of this video, I mentioned that DNA needs to undergo a process of replication, which generally takes place before cell division, before mitosis or before meiosis. And what we are going to do now is look at this process of DNA replication in detail. When it comes to DNA replication, like many other processes in biology, it is controlled by enzymes. So that's our first important point. We need to know all the enzymes and when they function and how they function. And secondly, the anti-parallel idea, the 5', 3' idea, also becomes extremely important in understanding the process of DNA replication. So the image on this slide shows the entire process holistically. However, we are going to break it down and focus on DNA replication step by step in order to understand it in sequence and logically. So let us start that. Okay, so when we have a look at this diagram, before we begin, let us just talk about some things first. Remember when we spoke about the 5' and 3' direction? So that becomes important in DNA replication, as I mentioned. So here we have our 5' end, and it ends in a 3' end. And that will be the complete opposite or the complete reverse for your complementary strand, which will run from 3' to 5'. And if you're confused about the 5' and the 3', have a look back at the structure of DNA and it will become a bit easier to understand. Furthermore, we know that adenine bonds to thymine and cytosine bonds to guanine, but they bond via hydrogen bonding, which is a very weak bond. So in black from the left, I'm just adding in a representation of these hydrogen bonds. And we can see that at some point those hydrogen bonds are no longer together anymore. They are weak. And that's where the process of DNA replication essentially starts from. It starts with an enzyme by the name of helicase. And this enzyme helicase unwinds your parental double-stranded helix DNA. So helicase acts along the parental strand and essentially splits that strand up into a sort of fork-like structure. There is then another enzyme called single-strand binding protein, which will be found on both sides of your parental DNA helix molecule, and that stabilizes these 2 strands. In other words, it keeps them open. So helicase opens the strands, and single-stranded binding proteins keep these strands open. We then have an enzyme by the name of topoisomerase, and topoisomerase simply rejoins the parental DNA. In other words, it connects those hydrogen bonds just before the replication fork. So here is your replication fork, and you can see just before the replication fork, topoisomerase rejoins some of those nucleotides from the opposite DNA templates, and that basically prevents the over-winding of the DNA molecule, to prevent your DNA molecule from breaking. Next, we have an enzyme by the name of primase, and primase is essentially, in everyday language, a type of glue, and that's a similar role to what primase does during DNA replication. So the enzyme primase has a different role on each strand. So we will focus on primase in the top strand, and then we will focus on primase in the bottom strand thereafter. So we can see that your primase adds on an RNA primer, which is this structure in red. Take a note that it is now in the 5' to 3' direction, that your RNA primer is added on to this strand, or to the top strand. So once again, we have our RNA primer, which is the structure in red. And once your RNA primer, which is like a glue, is added on to the top template strand, your next enzyme can play a role. And the next enzyme is called DNA polymerase 3. And DNA polymerase 3 adds on DNA nucleotides to the RNA primer 3' carbon. In other words, DNA polymerase 3 will add on DNA nucleotides in the 3' direction of your RNA primer. So as we can see here in green, I have circled your RNA primer. DNA polymerase 3 then moves down your strand, and as it moves down, it adds in DNA nucleotides, which are shown in light blue in this image, in the 3' direction of your RNA primer. And this process happens continuously, and it forms what is called the leading strand. However, when we have a look at the bottom strand, we see that there is a difference in our direction. Whereas in the top strand, we were able to move from the 5' to 3' direction of our RNA primer, we can't do that in our bottom strand. In other words, we can't form a continuous line of DNA nucleotides from DNA polymerase 3 in your bottom strand. And how that is solved is as follows. On the bottom strand, your RNA primase adds in segments of primers. And to these segments of primers, your DNA polymerase can then add in DNA nucleotides in fragments along the template. And these fragments have a special name, and the name of these little fragments is called Okazaki Fragments. It is a Japanese name by the Japanese scientist who discovered this process. However, we notice that we still have RNA in our DNA molecule, and we don't want that to be in our molecule. And that's where our next enzyme comes into play. That's DNA polymerase 1. And DNA polymerase 1 simply removes or takes out your RNA nucleotides and replaces them with DNA nucleotides. And all of your nucleotides are then glued together by your final enzyme, DNA ligase. So that is the process of DNA replication. To summarize it briefly, we have a parental strand. That parental strand is made up of two strands which are bonded together by hydrogen bonds. The enzyme helicase will unwind that strand. Single-stranded binded protein will help keep the strands open. Topoisomerase prevents your strand from overwinding just before the replication fork. We then have the leading strand, which is formed continuously, and we then have the lagging strand, which is formed in a series of fragments called Okazaki fragments. The reason why we have a leading strand and a lagging strand is because of the difference in 3' and 5' direction. In other words, because your DNA molecule is anti-parallel. Okay, and next we're going to move on to our last part of DNA, which is looking at biotechnology. And biotechnology is essentially the use of living organisms to make life a little bit more easier for humans, and that involves 4 different segments, starting off with cloning. So cloning can either be a tissue culture where we clone a certain group of cells, or we clone a certain tissue, or it can be embryonic where we clone an entire organism. Our second part of biotechnology looks at insulin production. So insulin is a hormone which converts your glucose in your blood into glycogen. And in some humans, their bodies can no longer do this because they've developed type 2 diabetes. And that's where the production of insulin becomes significant. We use the plasmid of DNA, and there's a process that we follow to produce insulin. We then have number 3, polymerase chain reaction, which is used in genetic profiling. And lastly, we have gel electrophoresis, which is used to produce a DNA fingerprint. So when it comes to DNA fingerprints, remember that we need to have 2 fingerprints to make a conclusion. So for example, if a crime was committed and we collect a sample of DNA and we create a DNA fingerprint, we can't determine who committed the crime. We need to identify certain suspects, get their DNA, and then create a profile or a DNA profile or a DNA fingerprint. And if that fingerprint matches to the one that we created originally, then we can have confirmation. So I've briefly undergone some of the processes of biotechnology. There's obviously a lot more depth and a lot more detail under them. However, I have just touched on them vaguely. And that brings our first unit of DNA to an end. And I hope you enjoyed it, and thank you so much for watching.